Hi, so I thought I would just do a video on the urea cycle, and then we're going to talk about protein degradation, or specifically the amino acid pool being broken down. So I'll just start here. So obviously the amino acids, they can be synthesized or they can come from the diet, and eventually they do need to be broken down via catabolism. So we have just a generic amino acid, which obviously has an NH3 plus group on it, and then we have an alpha ketoglutarate here. We're going to use an enzyme called aminotransferase. There's different kinds depending on the amino acid that you're working with. There's AST, which uses aspartate. There's ALT, which uses alanine. This is just a generic version. But just to note that all aminotransferases, they're going to need vitamin B6, which we abbreviate as PLP. And then as a result, you're going to combine your generic amino acid and your alpha ketoglutarate. You're going to get out an alpha keto acid and a glutamate, which is going to have the NH3+. So it's kind of like a hot potato, pass it on to the next guy. Alpha keto acid, this is just generic. It could be a pyruvate or it could be oxaloacetate. It just kind of depends on the configuration of the molecule. I believe that there's also gamma and there's also beta keto acids, but it's out of the scope, so we're not going to be concerned with it. So now we have our glutamate here with the NH3+ on it. We're going to put it into an enzyme called a glutamate dehydrogenase, which is a reversible reaction. Going in this direction, you're going to put in an NAD+, you're going to get out an NADH, and then we're going to actually rip off that NH3+, and that's going to join the urea cycle, which we'll talk about in a little bit. It is favorable to go in this direction. So if you see, it's removing, so this is going to be, this is not favorable by tacking on the H onto it, but removing this NH3+ is favorable. So it's going to be coupled in order to make it a favorable reaction. So most likely this would be negative delta G. And then with this reaction, it's going to be the reverse. So what we're going to do is we're going to be converting this alpha ketoglutarate into glutamate. And we're going to be adding on this charged particle, this ammonia, and then we're going to, it's not going to be favorable, so we couple it with this, and when the NADPH, and when we pluck that off, that creates enough energy to push the reaction in the forward direction, theoretically speaking. And so this should also be negative delta G. But that's out of the scope. I was just kind of mentioning it to tie things in. And so an activator for this would be a positive, ADP, and then an inhibitor would be a GTP. And this alpha ketoglutarate can join the TCA cycle, and then eventually it'll make some energy, which is pretty cool. So that's that whole mechanism there. So now I'm going to talk about the actual urea cycle. So up here we mentioned the amino acid pool. You have catabolism, and then as a result we get this NH3+. So where is this specific reaction occurring? This reaction is occurring in the liver specifically. This part is the mitochondria of the liver. So what happens is this NH3+ is going to go, it's toxic for the body. So what the liver does, which is pretty remarkable actually, is it transforms it into a not so toxic form, and then it can be excreted via the kidneys. So this NH3+ is going to combine with the CO2, and then we have this enzyme, it's called CPS1, there's a CPS2, which is a part of the pyrimidine synthesis, which we're not going to talk about in this video. So the CPS1 is going to take CO2, NH3+, and using 2 ATP, it's going to convert all of these, the ATP, it's going to convert CO2, NH3+, into carbamoyl phosphate. And then it's going to knock off, it's going to use those 2 ATP. This is a rate-limiting step. And it has an activator which I've abbreviated it as NAG. And basically you have something called a NAG synthase. And NAG synthase is going to take acetyl-CoA and it's going to take glutamate, and it's going to combine them to form NAG. And that NAG is going to be an activator for the CPS1. And then arginine is an activator for NAG synthase. And then so as a result, theoretically speaking, the arginine and the NAG, they're both going to get this reaction going. The arginine is specifically for the NAG synthase, and then the NAG is specifically for the CPS1. So those are the activators. We get this carbamoyl phosphate on it, and then there's something called ornithine, which happens a little bit later on in this cycle, but just trust me on it. So we get ornithine, and the ornithine and the carbamoyl phosphate are going to combine using an enzyme called OTC. The phosphate's going to come off of it, so that's going to make it a very favorable reaction, and then as a result, we're going to get citrulline. Now, we said that this is the mitochondria. This would be your cytosol, which are just the components within the cell, but not in the mitochondria. The citrulline can leave the mitochondria, enter the cytosol, and then I had mentioned previously these transamination reactions, and here is an example of one. So we have AST. So this is a reversible reaction, as I said previously, and the AST is going to need vitamin B6, the PLP. And what's going to happen here is it's going to take a glutamate and oxaloacetate, and it's going to put them through this enzyme, and you're going to get alpha-ketoglutarate, and you're going to get aspartate. So aspartate and citrulline are going to combine using an arginosuccinate synthase. That's going to create your arginosuccinate. Then we have another enzyme called arginosuccinate lyase, and that's going to just cleave things. So arginine is going to be an intermediate in this reaction, which is kind of interesting. But if you see here, the arginosuccinate lyase, it actually cleaves off a fumarate. Using fumarase, we can convert it to malate, and then malate dehydrogenase will then convert it to oxaloacetate, and then we can keep the whole thing going again. And then this step, which is super important, so arginine is going to go through an arginase, that's the enzyme, and it's actually going to produce the urea. The urea is then going to be transported to the kidneys, and then it'll be excreted via urination. The arginase also produces ornithine, and then there's another reaction that was previously mentioned in lecture. So this ornithine can be converted into a semialdehyde, and then a semialdehyde can become a glutamate. And then using either AST, ALT, or glutamate dehydrogenase, you can actually get alpha-ketoglutarate out of that. So that's kind of just like a little thing. And then again, this can go into the TCA cycle. Yeah, and I believe technically it could theoretically participate in this reaction as well, again, but that would kind of be a reverse of what it's doing there. And then the ornithine is able to go into the mitochondria, and then once it's in the mitochondria, the cycle repeats itself. So that's kind of just a broad overview of the mechanisms and how everything works. If you have any questions, feel free to leave me a question in the comments, or if I made a mistake, please let me know. But yeah, I hope you enjoyed the video. Thank you for watching.